Okay, the last one, the last method is the gene machine. The gene machine is now possible to manufacture genes in the lab in the following way. So the base of a gene can be worked out. So we can work out what are the bases of a gene. How? Because first of all we do, we see, working backwards, we get the protein and we see what is the primary structure of the protein, what are the sequences of amino acids. And if we know the sequence of amino acid, can we know the codons for them? Can we know the mRNA codons? We can, because we know which codon codes for which amino acid. So if we find the mRNA structure sequence, then we can go backwards to find the DNA triplet, the DNA code. So we can find the sequence of human gene. So what we do is very simple, is that we type into the machine the desired sequence of the gene. So we type in our sequence like A, T, T, C, C, G, G. So we put the desired sequence of the gene that we have. So it could be artificially made, it could be a known one for a human gene. Just put it in. The computer will check and will make sure that it's safe, there's no hazard, it will not cause harm. Then what it does is that the computer is going to construct something called oligonucleotides. Oligonucleotides. What's our purpose here? The whole purpose of this is to make an insulin gene, an actual gene. So we know what a gene is, right? It's made out of DNA, lots of DNA nucleotides. So we just typed in the basis, the sequence on the computer. The computer, in the machine, there are lots of nucleotides, DNA nucleotides. We just need to sequence them in the right order. So the computer does not make a long chain. What it does, it makes small chains called oligonucleotide. These are small strands of overlapping DNA. So it makes chunks of DNA, chunks of the gene, you can say. You can say it makes chunks of the gene called oligonucleotides. And all of them are made and then they are joined together. And that will make your insulin gene. So as you can see here, these are your oligonucleotides. Okay, there are small, so it's those sort of small chunks of the gene. And then all of them are joined together to make the whole massive gene. Now the good thing is there is no introns. Why? Because we are the one who are typing in the insulin gene. We can make sure that we don't type in any introns. So that's the good thing. Now we have our insulin gene or whatever desired gene that you want, and good thing is it has no introns, and we only have one copy first. Then you can use a machine called PCR, which you'll cover later on, and that PCR will amplify. Amplify means make more of this gene, and this gene can be used for our purpose. We have our gene here, and we basically can make a cut here and cut here, meaning the sticky ends, using which enzyme? Restriction endonucleases, and also the plasmid, same enzyme to make a cut, and then we can join them together to form our recombinant DNA.